Assalamualaikum warahmatullahi wabarakatuh. Today, we are going to present about our report on the experiment gas chromatography, separation and analysis of volatile organic compounds. I will start this presentation by stating the objectives for this experiment. First, to determine the retention time of N-butanol and 2-propanol. Second, to identify the components present in a standard mixture based on retention time. Third, to identify the components present in an unknown sample. And fourth, to determine the effect of temperature on retention time and resolution. Next, let's talk about the introduction to this experiment. Gas chromatography is an analytical technique for separating, identifying, and quantifying volatile compounds. It utilizes a stationary phase and a moving gas phase with various types distinguished by their detectors, enabling diverse applications in chemical analysis. Retention time in gas chromatography refers to the time taken for a specific analyte to travel through the chromatographic column and elute from the system, influenced by factors like the column dimensions, temperature gradient, and gas flow. It is crucial for identifying and quantifying compounds in complex mixtures, especially during comparative analysis. I will now pass the presentation to Ain, who will talk about the apparatus and chemicals.
For this experiment, several basic apparatus and chemicals were used. The apparatus that needed are a string, a beaker, and a dropper. As for the chemicals used, 2-propanol and butanol, a standard mixture of 2-propanol and N-butanol in a 1:1 ratio, and an unknown sample. Next, let's hear the methodology from Najihah.
Now, I would like to explain the methodology for this experiment. First, the string was rinsed before it was filled with the sample to prevent contamination. Then, the string was filled with more sample than the required volume. Next, the string was checked to make sure there were no air bubbles. Any air bubbles were removed by gently tapping the string. After that, the string was held vertically with the needle pointing upward. The plunger was slowly pushed until the required volume was reached at the eye level. The excess sample was removed using a tissue. Once the sample was prepared, it was injected into the GC injector. In the GC machine, the carrier gas flowed from the gas cylinder through the pressure regulator and flow controller. The carrier gas carried the injected sample into the GC column located inside the oven. Inside the column, the sample components were separated based on their properties. After separation, the components entered the detector, which in this experiment was a flame ionization detector or FID. Finally, the detector sent signals to the computer and the results were displayed as a chromatogram analysis.
So first, data analysis and result. This chromatogram shows the result for pure 2-propanol analyzed at 70 degrees Celsius. A single shot is observed at the retention time of approximately 1.56 minutes. This indicates the samples is pure and well separated with no significant impurities. Next, this slide shows a chromatogram for N-butanol at 70 degrees Celsius. The main peak appears at the retention time of approximately 1.95 minutes, which is longer than 2-propanol. This is due to N-butanol having a high boiling point and lower volatility. For the 1:1 mixture analyzed at 70 degrees Celsius, two distinct peaks are observed. The first peak corresponds to 2-propanol, while the second peak corresponds to N-butanol. This confirms that the gas chromatography system successfully separates both volatile compounds in the mixture. Next, the chromatogram represents the unknown sample with a 2:1 ratio. Two peaks are observed at retention time similar to those of 2-propanol and N-butanol. Based on the peak height and area, the peak at around 1.56 minutes is larger, indicating that 2-propanol is a major component in the unknown sample. At 100 degrees Celsius, the standard mixture shows shorter retention time compared to 70 degrees Celsius. This indicates faster elution of compounds to increase temperature. However, two peaks are still clearly separate, showing acceptable resolution. For 150 degrees Celsius, retention time are further reduced. This peak elute much faster, but distance between peaks are smaller. This suggests that although higher temperature speed up analysis, it can reduce separation efficiency. And lastly, overall, the results show that retention time depends on compound volatility and column temperature. So 2-propanol consistently elutes before n-butanol due to its lower boiling point, increased temperature, reducing retention time, but may compromise resolutions.
Now, I will answer the questions based on this experiment. First, state the types of compounds which are suitable for analysis using GC. The types of compounds suitable for analysis using gas chromatography are volatile compounds, thermally stable substances, and low molecular weight organic compounds. Examples of these compounds include hydrocarbons such as hexane and octane, alcohols like pentanol and butanol, and ketones such as acetone. Next, why is FID a suitable detector for this analysis? FID is a suitable detector for this analysis because it is highly sensitive and has a wide dynamic range. The signal produced depends on the number of carbon atoms in the organic analyte, making it suitable for organic compound analysis. In addition, FID is mass sensitive rather than concentration sensitive. Lastly, list two factors which can increase the efficiency of a GC column. Two factors that can increase the efficiency of a GC column are the volatility of the compound and the length of the column. Next, let's hear discussion from Putri.
For this experiment, gas chromatography was used as an analytical technique to separate volatile compounds based on their boiling points and their interaction with the stationary phase inside the column. In gas chromatography, compounds with the lower boiling point are more volatile and interact less strongly with the stationary phase. As a result, they travel faster through the column and have shorter retention time. On the other hand, compounds with a higher boiling point or stronger interaction with stationary phase move more slowly and therefore show a longer retention time. Based on the data shown at 70 degrees Celsius, 2-propanol, which has a lower boiling point of 82.3 degrees Celsius, elutes earlier with a shorter retention time of about 1.559 minutes. Meanwhile, n-butanol with a higher boiling point of 117.7 degrees Celsius show a longer retention time of 1.954 minutes. This result clearly demonstrates the relationship between boiling point and retention time in gas chromatography. Next, we analyze the chromatogram of the unknown sample at 70 degrees Celsius. Two peaks were observed at retention time of 1.557 minutes and 1.919 minutes. By comparing these values with the standard mixture, the peak at 1.557 minutes correspond to 2-propanol, while the peak at 1.919 minutes correspond to n-butanol. It can also be observed that 2-propanol peak is larger than the n-butanol peak. This indicates that the relative amount of 2-propanol present in the unknown mixture is higher. Based on the peak size, the result quantitatively suggests that the unknown sample is approximately 2:1 ratio of 2-propanol to n-butanol. Lastly, the effect of column temperature on retention times was studied. As shown in the table, when the column temperature increases from 70 to 100 and 140 degrees Celsius, the retention times for all compounds decrease. This occurs because higher temperature increase the volatility of the compound and reduce their interaction with the stationary phase, allowing them to pass through the column more quickly. In addition to temperature, molecular structure also plays an important role in separation. Smaller and less polar molecules generally experience weaker interaction with the stationary phase and move faster through the column. In contrast, larger or more polar molecules interact more strongly, resulting in longer retention times. Overall, these results confirm that both boiling point and temperature significantly influence the separation efficiency in gas chromatography. Now, we move on to IU for the conclusion.
Gas chromatography successfully separated and identified volatile organic compounds based on their retention times. 2-propanol elute early than n-butanol due to its higher volatility. Increasing temperature reduced retention time, but slightly decreased peak resolutions. So that's all for this. Thank you.